Hello, I'm Jan Leifman.
I'm Gayatri Pramal Menon.
Welcome. Welcome to MedNews Week Conference, where we feature presentations by medicine's global leaders. Today, we have an amazing keynote speaker in Dr. Leonard Zon. Dr. Zon is the director of the stem cell program at Boston Children's Hospital.
In today's presentation, Dr. Zon discusses the latest in stem cell clonality.
Did you know that Dr. Zon is an internationally recognized physician, leader, and pioneer in stem cell biology, cancer genetics, and hematology? He has defined 5 new human diseases as a result of first identifying the genetic basis in a zebrafish mutant. He discovered a small molecule called prostaglandin E2 that regulates stem cell biology that was translated into the clinic for enhancing hematopoietic stem cell engraftment.
He's a dedicated advocate and a leader in the field of oncology and clinical trials.
So let's tune in. Let's tune in and learn from this great global leader.
So most of my career, I've been working on blood stem cells or hematopoietic stem cells. If you think about the bones, inside the bones is the marrow, and that is the place where blood cells are formed. There's a constant influx and efflux of hematopoietic stem cells. These stem cells have the ability to self-renew, or they can differentiate into the blood lineages. This process is facilitated by the niche cells, such as the endothelial cells and the stromal cells. And I'm going to talk a lot about the niche cells and how they help this process. Blood stem cells is the only stem cell with a quantitative assay. That's a competitive repopulation assay. It's the only stem cell besides skin that's used therapeutically. And we know that bone marrow transplants, thousands of bone marrow transplants are done every year, and hundreds of gene therapy and gene editing trials use blood stem cells. And when a mutation happens in a blood stem cell, it can lead to clonal expansion and ultimately leukemia, and I'll talk about that also. Now, my preference of a model system to study this is a little bit unusual. It's the zebrafish. And so zebrafish are wonderful because the embryos are completely transparent and they make circulating blood in 23 hours. And over the years, we've made transgenic fish that light up every single blood cell in a different color. Each mother has between 100 to 200 babies every week, so it's a fantastic genetic system. And it's possible to use genetic screens or gene knockdowns to test gene function. We can add chemicals to the water and cause phenotypes to occur, and the blood program is amazingly conserved. I want to show you a movie of how blood stem cells are made. And shown here is a 36-hour embryo. And in all vertebrates, the blood stem cells are actually made in the dorsal aorta, which is your largest blood vessel. So we're going to dive into the aorta, and what we find is that an endothelial cell actually changes its fate. It rounds up to become a blood stem cell. That cell could be transplanted, but endogenously, it goes off into circulation. And it's going to go around and around circulation and land in the tail of the zebrafish in a place where blood cells are forming. This is very similar to the fetal liver of a human. The cell will come in and transmigrate into the niche, and this will double the stem cells. And then some of these stem cells will go into circulation to colonize the marrow. In the zebrafish, the marrow is in its kidney and making the kidney a hematopoietic organ. But in the human, it would be in the bones, in the bone marrow. Sometimes the cells bypass the kidney and go to the thymus, and this will start the immune system. And all of this allows this animal to have blood really throughout its entire lifetime. Now, several years ago, we were able to make a fish that had green stem cells. We used an enhancer from a specific gene, RUNX1, and it turned the stem cells green. And we could see the stem cells are born in the aorta, they go round and around circulation, and they land. Now, I want to show you what happens when a stem cell lands, and you could think of this even as a transplanted stem cell. So focus on this circle. What we see here is that the stem cell transmigrates outside the blood vessels. But then we saw that the blood vessels wrap themselves back around the stem cell in a process we call endothelial cuddling. Then the stem cell divides and one stem cell leaves and the other one stays. And this is a very stereotypic process. So those endothelial cells cuddling are very, very important. We took this very animal and we did correlative electron microscopy. And we found some surprises. So first of all, the stem cell that had been circulating finds another cell to attach to, a stromal cell. And the divisions of the stem cell are perpendicular to the plane of the stromal cell. Now, we also know that the stem cell is surrounded by 5 blood vessel cells, by 5 endothelial cells. And this forms the niche. So we wanted to understand more about this particular niche. And one of the things that we've done recently is RNA tomography. This is a really interesting technique called tomoseq. We section across the embryo, sectioning through the stem cell niche. And then every section goes into a tube for RNA-seq. So you tell all the genes in each one of these sections. And what we're able to find are these genes here that are adjacent, right next to the stem cells. And so we were interested in what kinds of genes, what are the cells, and in particular, we were interested in the endothelial cells. So 29 of the genes were endothelial specific, blood vessel specific. And we can do a technique called in situ hybridization. And if you look at these genes, they're only next to where the stem cells are in this tail region. And that's really amazing. And it made me wonder, what are these endothelial cells and how do they actually function? And these endothelial cells are veins, but they're specialized veins called venous sinusoids. So one of the things that we did was to take the promoter of 2 of those genes, E-selectin or this mannose receptor, and we drove green fluorescent protein in a fish there where all their blood vessels are red. And you can see here that you only see expression just in this little part here where the venous sinusoids are. So this is a mannose receptor, and it says somehow this 1.3 kilobase of DNA has the information to make those venous sinusoids express. So what we did was we used a technique called ATAC-seq. And ATAC-seq allows you to look at open chromatin. And we could see that there's open chromatin just in the venous sinusoids, but not in other endothelial cells. And this little peak here is about 125 base pairs of DNA. So we took that 125 base pairs and drove green fluorescent protein. And again, only did you get this particular set of tissue lighting up. So what we realized by looking at all the peaks that were venous sinusoid specific is they were enriched for several sites for DNA binding proteins, ETS, SOX, and a nuclear hormone receptor. And we eventually found transcription factors, DNA binding proteins, that could actually bind to those motifs. And we could use these 3 transcription factors to actually reprogram ectopic niches in vivo. And I'll show you that work now. So first of all, when you look at the peaks that are only in the venous sinusoids, but not in the other ones, and then you take these peaks and drive green fluorescent protein, you only get it in these tail regions. And so this seems like there's very good information there. When we look at the sequences, again, we see the ETS, SOX, and nuclear hormone receptor sites. So we think that these sites are the sites that confer a fate to a cell. So we knew the sites were there, and we looked at our RNA-seq expression, and we could see these transcription factors, these 7 transcription factors that could participate in binding to those motifs. And so we did an experiment that's very similar to a friend of mine, Shinya Yamanaka, who won the Nobel Prize for reprogramming. We overexpressed these these 7 factors, I should say. And when we did that, what you could see is compared to, let's say, this embryo here, which is a control, when we overexpress the 7 transcription factors, we now get new blood vessels. And you can actually see the blood vessels with our fluorescent reporter. Now, we were able to find, again, this combination, ETB2, SOX7, and NR2F2, and that led to a very sizable number of animals having ectopic vessels, new vessels. So then my postdoc, Elliot Hagedorn, came to me and said, this is the best day I've ever had as a postdoc. And what he found was he did put all 3 transcription factors into an embryo that had green blood vessels, green venous sinusoids, and red stem cells. And what he found was that the stem cells left their usual position and went to the new vessels. You can see that here. And then they did this cuddling behavior also. And then what we're able to demonstrate now, and focus on this square, which is the movie. Here are the normal niche with the number of stem cells there. But here you have a new niche. So let's picture it as like a new marrow in a place where it shouldn't really be. But because we've added these 3 transcription factors, it's reprogrammed cells to take on this fate. So follow this here in this movie. What you can see is that the stem cells are dividing in that niche. So we've really fully reconstructed a new niche, and this is very, very exciting. The genes that are expressed in the embryo turn out to be the exact same genes that were found in the adult marrow. And then we had a friend who had taken out the endothelial cells of a mouse, and we found that during fetal life, our signature of genes was very high in the liver. And remember, the fetal liver in a human or in a mouse is the site of blood formation. And then later in an adult, you can see the bone marrow has very high expression. So we've been able to take these genes, find venous sinusoid-specific enhancers, find the DNA binding proteins or transcription factors that bind to those motifs, and use those actually to reprogram an ectopic niche, an ectopic marrow. Now, what would you use this for? Well, it turns out there's a disease called myelofibrosis. In this disease, the entire marrow becomes fibrotic. The stem cells can't stay there, so they leave and go off into circulation, and they try to colonize the liver and the spleen in a process called extramedullary hematopoiesis. But this is very inefficient. And perhaps we could actually take human iPS cells and reprogram them to make these veins. Then we could inject them into the arm or maybe into the liver, and they would make a new site for blood cells to be forming. And we've actually succeeded in doing this with the human iPS cells, and we'd really like to translate this into humans at this point. So now I'm going to spend the remaining part of my time on talking about clonality. So one of the things that we can do in the hematopoietic system is to do a marrow transplant. So when we do that in any animal, including humans, we can take out the marrow and we transplant into an irradiated host or a host that's received a lot of chemotherapy. But when we're interested in talking about clones of stem cells, it's very hard in this system because the host niche is inflamed, the transplanted cells are really stressed, and only some clones contribute after the transplant. So a group of us have started doing a technique called in vivo cellular barcoding. And this is very exciting. I'm going to show you the work in the fish, but we can also do this in humans. So we have a technique called Zebrabow and another one called Gestalt. I'll talk about these. And all you do is bleed the animal monthly and do a marrow analysis, and you'll be able to look at the clones of stem cells. Here, there's no transplant, there's no stress. The stem cells are in their native environment. And this is an accurate assessment of clonality. So why did we want to study clones? Well, it turns out that this is one of the hottest topics in all of biology. So what goes on is that you have hematopoietic stem cells, and they are polyclonal. In other words, many hematopoietic stem cells make the cells of your blood. But it turns out as we age, many humans develop mutations in epigenetic regulators, DNMT3A, TET2, and ASXL1. And this causes a stem cell to occupy up to 20 or 30% of the peripheral blood. This is a syndrome called clonal hematopoiesis of indeterminate potential, or CHIP. These cells can go on to become myelodysplasia and also into leukemia. Now, the amazing part about this condition is it's very, very common. 13% of all 70-year-olds have clonal hematopoiesis. In other words, their stem cells are mutated. 30% of 80-year-olds and 50% of 90-year-olds. Now, the other thing is this condition is inflammatory. And so people with this end up with blood clots, heart attacks, and strokes. And also we found recently cirrhosis and even kidney disease. So how do we study clones? Well, in this system, the Zebrafish system, we're able to mark each stem cell in a different color. The way we do this is we have 15 to 20 copies of this plasmid throughout the entire genome. And it's red, blue, and green, separated by recombination sites. When you express a blood-specific CRE, which is the recombinase, you start to get lots of different colors. And all of the embryonic blood is different colors. Now, embryonic blood is really transient. And so essentially, as the animal develops and grows to adulthood, all the embryonic blood dies off, but the stem cells that are colored will end up contributing to the peripheral blood. And you can use the colors in the peripheral blood to back calculate how many stem cells were born in the aorta. So here's an animal that has multicolored blood. It's pretty cool, huh? And you can see here, this cell here, which is in an extravascular location and just divided, show you that again right there, that's most likely a stem cell. So that blue-green cell, when it grows up to adulthood, will make blue-green blood. So now we're going to grow the fish up to adulthood and bleed it. And this is what the peripheral blood looks like. Now, I'm a hematologist, and I take care of kids with blood diseases and cancer. And I can tell you, this is the prettiest blood smear I've ever seen. Now, when you back calculate how many stem cells you get, at 24 hours, which is just before the birth of the blood stem cells, there are 21 clones of stem cells. So now let's use this system to study clonal hematopoiesis. So first of all, many laboratories have shown that the mutant stem cell makes mutant neutrophils and macrophages pretty well. But these macrophages and neutrophils pump out inflammatory cytokines. They're making interleukin-1 and TNF-alpha. This makes the entire marrow inflamed. Then, by an unknown process, there's clonal dominance and pre-leukemia. So I want to tell you how we've solved what happens here. So we invented a technique called twister, or tissue editing with inducible stem cell tagging via recombination. It's actually a simple concept. What we did is we injected guide RNAs for the 20 genes that cause clonal hematopoiesis in humans. And we use CRISPR-Cas9 to actually do the knockouts of these genes. But we titrate down the amount of Cas9, so 50% of the stem cells are normal and 50% are mutant. So they can compete in vivo and see who wins. And then we just bleed the fish every month or so, seeing what the colors are to look at clonal dominance. And then at the end, we may take out the marrow and sort the dominant clone. And what's nice about this color system is that this dominant clone is one color. So you can really study it very, very well. So here's a control, and you get polyclonal hematopoiesis. Many different colors contribute to blood. But here's a mutant pool, and you can see this huge clone here. 60% of the blood comes from one stem cell, and it's that green stem cell. We see this as clonal dominance. The mutant pool gives a higher number in terms of cluster size. And you can see an economics term, Gini coefficient, also is higher in the mutant pool, meaning that there's been a clonal dominance and certain colors are being dominant. The driver in our system that's most potent is a gene called ASXL1. It's an epigenetic regulator. It's only exon 12 that has the mutations, and exon 12 in humans is the one that causes the dominance. So what did we find? Well, we found that the mutant stem cells make the neutrophils, and these neutrophils are making IL-1 and TNF-alpha, as well as the macrophages. So these inflammatory cytokines are being expressed in our model from the mutant neutrophils and macrophages. But the thing we found, which is really exciting, is the stem cells actually are making anti-inflammatory factors, SOX3A, NR4A1, and ATF3. So this provides the cell a survival advantage in the time of the stress from these inflammatory cytokines. And that's why it takes time for this to occur and why older people have a clonal dominance. So what we did to prove this was we knocked out just ASXL1 by itself. This led to clonal dominance. But we also knocked out ASXL1 and NR4A1. Our hypothesis was that this should suppress the clonal dominance. And that's what we saw. So over here, if you look at the NR4A1 homozygous mutants, we saw that these were lower than the rest. And so this shows that you restore normal balance if you knock out NR4A1 by CRISPR. Now, NR4A1 deficiency had no effect on normal stem cells. So this could actually be a therapeutic. And so what we've been able to show in our twister system is that the mutant stem cells make mutant macrophages and neutrophils. They look normal, but they're pumping out inflammatory cytokines. This leads to upregulation of NR4A1 specifically in the mutant population. And then this leads to clonal fitness, and you get the expansion over time. But if you knock out NR4A1, then you reduce the clone size, indicating that it could be a therapeutic. Now, what's exciting is recently we found a tool compound that's an NR4A1 antagonist, because this is a nuclear hormone receptor. And we've been able to show in our FISH system that this lowers the variant allele fractions of ASXL1, meaning it could be a therapeutic. So now, in collaboration with some colleagues, we're trying to show that this works on human cells. And then if that's successful, it could lead to a new therapeutic. So I want to finish now with a story about macrophages. And this is about a very new process, a quality assurance mechanism of blood stem cells. So this started off with this really fascinating movie. These are macrophages, and they're interacting with green stem cells. And I hope you can see that they're actually removing material from the stem cell. And so this intimate interaction was just really exciting. We didn't understand why these macrophages were interacting with stem cells. And Sam Watrous studied this process. So what he found, which is very surprising, is after the birth of blood stem cells, about 20% of all of the stem cells are completely eaten. But then almost every other stem cell interacts with a macrophage. And when we see the interaction, the macrophage comes in, removes a little bit of the stem cell, the cytoplasm, and then 45 minutes later, the stem cell will divide. And this is very stereotypic. So all of this suggests some type of quality assurance mechanism that the macrophages are patrolling and looking at the stem cells and seeing if they're a good stem cell or a bad stem cell. So in the zebrafish, we can get rid of macrophages very easily, either using a drug, clodronate liposomes, or we can use IRF8, a gene that when you knock it out, reduces macrophages. So what we did is we either did the IRF8 morpholino or we gave clodronate, and we did this with our rainbow fish, with the twister system. And what we saw was really remarkable, that if you eliminate the macrophages, you get half the number of stem cell clones in adulthood. So these macrophages really are patrolling and trying to determine which are the best stem cells. So we tried to understand the process at a molecular level. One of the things we found is that the green macrophages that ate the red stem cells would keep the red material inside them, allowing us to FACS sort these cells and being able to study them. So first we did single cell RNA-seq to be able to look at this populations, and the macrophages that were eating the stem cells were this particular population, number 3. And that was associated with phagocytosis. And you could see these have lysosomes and cholesterol transport. And one marker of these macrophages is HMOX1. Now, many of you may know about macrophage populations. There's M1 and M2. And actually now there's probably 20 different macrophage populations. And this is one that seems to be responding to oxidative stress. The thing that really helped us was to study proteomics. And we did a technique called few cell proteomics. We could do proteomics on 120 cells, and we studied the macrophages that had eaten the stem cells versus the macrophages that hadn't. So what we found was very exciting. We found 3 paralogs of calreticulin that were enriched in these cells that were eating the stem cells. So what is calreticulin? Well, calreticulin is an endoplasmic reticular protein. It has a retention signal called KDEL. In times of stress, calreticulin can go to the surface of stem cells and actually bind there. And there it can become an eat-me signal. And this was pioneered by the Weissman laboratory, where he found on normal cells, or particularly in cancer cells, calreticulin could be an eat-me signal by interacting with the LRP1 receptor on macrophages. And our macrophages have very high levels of LRP1. So we were able to take a mutant calreticulin that lacked the KDEL sequence, so it would get to the surface very easily. And this made us have many more macrophage stem cell interactions. And you can see that here, any of the different CalR isoforms, CalR, CalR3A, and 3B, all led to this phenotype. But then we did something really cool. We knocked down calreticulin paralogs, and we studied them for macrophage stem cell interactions by imaging, and then used the Brainbow system to look at clonality. So what you can see here is that when you knock down CalR3A and 3B, you get a reduction in the number of stem cell macrophage interactions, and you can see clonality is reduced by CalR deficiency. But we also did something you can do in zebrafish. We made a parabiotic fish. We fused 2 fish together. They end up joining their circulations, and we can look at whether the stem cell or the macrophage requires the calreticulin. So shown here is, on this side, is you have red stem cells with the CalR knocked down. On this side, you just have green macrophages. Under this particular configuration, you get less macrophage stem cell interactions. And that's just quantified here, that if you knock down on the stem cell side, the calreticulin, you reduce the interactions. But if you knock down on the macrophage side, you don't reduce the interactions. So it's the calreticulin on the stem cell that's responsible. Now we could see that these macrophages love to interact with stem cells when they're in the G2M phase. And we do this with a fancy fish called Fuchi that only lights up in the G2M phase. And then we're able to label the cell divisions. And you can see that, again, they love these ones when they're in G2M. And it takes about 45 minutes for the stem cells to divide. And we're also able to use EDU labeling to look at the stem cells. And when we do that, we can see, again, less cycling stem cells if they are not interacting with macrophages. So one of the things that we realized is macrophages are a source of IL-1, interleukin-1. And we're able to demonstrate that if you take the IRF8 morpholino, which has reduced macrophages, but if we add in IL-1, we rescue back those cycling stem cells. And this is dependent on the MAP kinase pathway that's downstream. But actually, a reviewer asked us to find out if macrophages deficient in IL-1 did the same thing as macrophages that had IL-1. And we realized we could do this using the parabiotic system. So in this system, what we did is we had a fish with blue macrophages, and we did an IL-1 knockdown in those fish. Over here, we had green macrophages that had normal levels of IL-1. And this allowed us to look at the animals at the cells that had the IL-1 or didn't. And in this beautiful parabiotic fish, what you can see is that the IL-1 knockdown macrophages did not allow the stem cells to proliferate. So it was a very, very cool result. So all of this suggested that the stem cells were being quality assured by the macrophages. But what is the macrophage sensing? Well, we studied our IRF8 knockdown stem cells, and what we found was there was a population here that had very high level of oxidative phosphorylation and FOXO signaling. And this suggested that the stem cells had reactive oxygen species. And we're able to knock down FOXO-A3, which will increase the amount of reactive oxygen species. And you can see more CALR interactions if that happens. And we saw more ROS in those cells. We also could use an antioxidant and prevent the interactions of the stem cells. So if you think about it, the oxidant stress is one of the perturbing things that actually leads to stem cells and macrophage interact. We also looked in our fish. Again, you could see that with CALR, there was more reactive oxygen species. And when we actually used an antioxidant, this reduced it. And it also reduced the number of RUNX cells. And we're able to actually look at the antioxidants and see, again, the fraction of interacting cells was reduced when you use those antioxidants. So the way to think about this is based on this movie. Here we have RUNX cell without ROS. And then over here, we have a RUNX cell that has ROS. So what we see with this one is that if there's no ROS, we see that these are the ones that lead to division. Whereas when we see ROS, reactive oxygen species, these are the ones that kill, doom the stem cell. So just in the last little bit, I'd like to talk about our recent work. And what we've done is a chemical genetic screen, because we'd love to be able to manipulate stem cell clones. And maybe we could get the macrophages to actually eat clones that are myelodysplastic or clones that are starting to develop leukemia. So what we did is we used immunostaining for surface calreticulin. We also set up something called a split turbo ID. We ended up getting the exact same result with these. And then we stained for reactive oxygen species. And we screened about 1,200 compounds, and we ended up with about 93 compounds that induced surface calreticulin. And 22 of those led to a macrophage stem cell interaction in the zebrafish. So 22 of these gave in vivo activity. So what are these 22 compounds? Well, one of the things that we found is the ones that lead to the reactive oxygen species If you look here at the orange is dooming and the grooming is black, well, these compounds give you a lot more dooming. These all lead to mitochondrial potential. And so we think these are leading to a lot of ROS, and that's making the cell get eaten completely. Now, some of these chemicals are giving grooming, and some of them don't even induce any ROS at all. And so we're now chasing what the grooming behavior is, trying to understand how that allows clones to actually proliferate. So the mitochondria is very important in this, and we started to study different mutants that either increase ROS or decrease ROS. And what we're able to demonstrate is the ones that lead to a very high ROS state, they will give you more dooming than grooming. But if you look at these that reduce the ROS, now you get more of the grooming behavior. And this is just one of the molecules, SOD2, which you can see here is going to lead to higher ROS, higher surface calreticulin, and more dooming events. So this leaves us with a quality assurance mechanism for macrophages with stem cells. And I'm going to take a little bit of time talking about this. Our motto would be is that if the stem cell has reactive oxygen species, and if it has a lot of this, then this is going to lead to a dooming behavior. This leads to high levels of surface calreticulin on the surface of the stem cell, a lot of interactions with macrophages, and ultimately the stem cell will be completely eaten. So it must be bad in evolution to have your stem cell have high reactive oxygen species. And probably this is a result of the trauma of being born as a stem cell and actually having to go through circulation to get to the next niche. Now, if you have an intermediate level of calreticulin, now you will still facilitate this interaction. And what's interesting about this situation is the macrophage that comes in is able to donate some IL-1, some interleukin-1, and through the MAP kinase pathway, this leads to the divisions of those stem cells. And then what you have are more of these cells, so they will contribute better to adulthood later on in life. So this is another way of thinking about these macrophage stem cell interactions. So here's this little stem cell that I showed you before, and it's going to go off into circulation. We think this is a stressful time in a stem cell's life. Okay, so now it's going to come around into that niche that I showed on one of the first movies of this, and we're going to look, and the stem cell is actually attaching to the endothelial wall and going to transmigrate now into the niche. Now, what we didn't know until now is a macrophage comes in and probes that stem cell. In this particular case, it completely ate the stem cell, so there must be something wrong with that stem cell. We think it has high ROS. But another behavior is that this macrophage is going to come in, and now it just takes a little bite of stem cell. There it goes. And now what it does is donate some interleukin-1. And now what happens is that stem cell is actually going to attach to a stromal cell over here. And then that stem cell will divide. And that's how the divisions will ultimately enrich these populations.